there. We here at the happiest place on earth have noticed an unsettling trend. More and more apathetic teens are failing to feel our special brand of magic. Isn't that right, Bobby?
Whatever.
Whatever indeed. But never fear, our Fungineers have developed a new gene therapy based on our new gene, magic. One quick injection into the ocular cavity and you too can have a vacation free of whining and emo BS. Let's hear more about our genes, shall we, Billy?
It's Bobby.
Whatever.
We all know that DNA codes for our genes, but what you may not know is that DNA gets transcribed into RNA before finally being translated into a protein. Furthermore, transcription is regulated by all kinds of factors and elements. There's distal elements, proximal elements, and core promoter elements. Our gene looks something like this. Although transcription starts in the +1 start site, there are many elements and transcription factors that affect its efficacy. Many of these elements are found in the core promoter region, but other elements are also found in the proximal promoter region and the distal promoter region. As you can see, the core promoter region is located between -40 base pairs and +40 base pairs. The proximal promoter region is located between -200 base pairs and approximately -40 base pairs, and the distal promoter region is located between upstream base pairs and -200 base pairs. The distal promoter region often includes locus control regions, which are composed of many elements that regulate several genes that are related to each other, both spatially and temporally, to coordinate their expression in either a tissue-specific or time-dependent manner. Let's look at each of these regions individually. In this configuration, our distal regulatory promoter region will be inhibiting transcription. When 5-5IAA, a metabolite of serotonin, binds to the silencer, it silences the activity of the gene. Furthermore, when the insulator is unmethylated, CTCF can bind, blocking the activity of the enhancer on the downstream promoter region. This is our distal regulatory promoter region. It has a silencer, an enhancer, and an insulator. At this time, you can see that it's actually set up to enhance transcription. POMC, which is a precursor to melatonin, is bound to our enhancer, and our insulator is methylated to prevent the binding of CTCF. In presence of high sunlight, POMC is produced and will bind to the enhancer, prompting a conformational change that you'll see when we talk about the PIC. Our magic gene is part of the larger locus control region, in which the magic gene is followed by genes in the dopaminergic system. We have engineered it so that it will be methylated in the presence of sunshine, so that the enhancer can not just affect our magic gene downstream, but also genes in the dopaminergic system, making it more pleasurable to attend Disney World when the sun is shining. Here, this is our proximal regulatory promoter region of our magic gene. Its motif includes a GC box, a glucocorticoid response element, and a cat box. In order for transcription to operate at maximum efficacy, all of these need to be bound to their transcription factors. The GC box needs to be bound to SP1. The GRE element needs to be bound to glucocorticoid receptors, which are in turn bound to cortisol. And the cat box needs to be bound to the cat box binding factor. Because high stress increases the amount of cortisol in serum, high stress levels can help increase the transcription rate of this particular gene. This is the core promoter region of our magic gene. You can see that its motif includes a B recognition element, a sequence-specific TATA box, another B recognition element, the initiator element, downstream promoter element. This is where our primary initiation complex will form and interact with mediator as well as enhancing elements to form the transcription machinery and get maximum transcription. Now let's look at how the pre-initiation complex forms in our core promoter region. First, TF2D attaches to the sequence-specific area of the TATA binding box. Next, TF2A attaches to the TATA F2D complex to stabilize. Then TF2B attaches to the BRE units. And is critical for the formation of a stable tertiary complex with the FTF2D. Next, TF binds with RNAP2 and recruits it over. On the core promoter, stabilizing the PIC, mediator complex, and TF2H and TF2E, are recruited to the PIC, and the formation of the open promoter is mediated by TFH. This is the general transcription machinery, but it can be enhanced when looping occurs and enhancing elements interact with mediator to maximize the transcription process. The looping process is further facilitated by cohesin. For our magic gene, we have POMC binding to an enhancing element. This maximizes the efficacy of the transcription pre-initiation complex formation process. Here we have an example of the looping that occurs when the enhancement element binds to its transcription factor, in this case POMC, which is a precursor to melatonin. The bound enhancer element interacts with mediator, which in turn interacts with RNAP and general transcription factors in the core promoter region. This looping is further facilitated by cohesin. You can see that now the enhancement element is in close proximity of the promoter region, where it can elicit a stronger transcription response for the magic gene. After the primary initiation complex has been formed, there are 3 stages to transcription initiation. An abortive initiation, transcripts less than 5 nucleotides long that are unstable are produced. Next, in promoter escape, transcripts of approximately 10 nucleotides long are produced. These favor promoter escape over abortive initiation. During this time, mediator places RNAP CTD terminal in close proximity to the TF2H, which phosphorylates the CTD. After promoter escape, productive initiation begins. This is when transcripts of approximately 25 nucleotides long are produced that signal elongation. The 5-cap structure is added to the 5' end of the RNA after these 25 nucleotide long products are produced. when the capping enzyme recognizes the phosphorylated CTD terminal.
Assembly of PIC. Transcription factor 2D binds to the TATA binding protein. Transcription factor 2A stabilizes the interaction between TF2D and the DNA. Transcription factor 2B binds to the recognition elements on DNA. Transcription factor 2F binds to the RNA polymerase II recruit to the core promoter and stabilizes the PIC. The C-terminal domain is on the RNA polymerase II and dephosphorylated, leading to the closed promoter complex. Transcription factor 2H and 2E join the PIC with TF2E, recruiting TF2H. TF2H is able to phosphorylate the C-terminal domain using the kinase of the TF2H, leading to the open promoter complex. This is the initiation of RNA synthesis. RNA polymerase II begins at the start site, and initiation occurs when phosphodiesterase bond is catalyzed via RNA polymerase II between the first two nucleotides. Abortive initiation occurs in the first 5 to 10 nucleotides and RNA polymerase restarts. C-terminal domain is phosphorylated by TF2H due to close proximity due to the mediator. The promoter is cleared once the transcription factors leave the complex, allowing RNA polymerase to continue initiation.
This is a replication of the transcription bubble. The yellow strand is the coding strand, and it's in the 5' to 3' direction. The red strand is the template strand. It is in the 3' to 5' direction. This is a histone, and just after the histone is the opening up of the DNA through melting. This is another histone as the DNA resumes its structure. RNAP2 will move along the template strand, adding nucleotides. And growing the nascent strand, adding nucleotides in the 3' orientation and growing the strand in the 5' orientation. In summary, magic gene expression happens when serotonin metabolites are low, CORT is high, POMC is high. It's part of a larger locus control region that involves the dopaminergic system and also the spendthrift gene. When these work together, people derive maximum pleasure out of their trips to Disney World, spend the maximum amount of money, and have the maximum desire to return again. Expression of this gene can take your sulky teen from this to this.
Well, Bobby, how was your trip to the happiest place on earth?
I give it 2 ears up.
Side effects may include delusions, hallucinations, thinking you can fly, and whatever that is. Oh.